to continue on this CMC talk, I'm curious how you think companies should strategize their approach to CMC, considering how much is changing, even just with the capability and components in these molecules. I mean, we talked about linker advancements and payload advancements. I guess where are the biggest gaps that teams could fall into, maybe in terms of developability, whether there's gaps in certain analytics or just hazards to be wary of as we're charting this relatively uncharted territory.
Yeah, I'll add some comments, and then Morris and Boris want to follow up. Go ahead. I mean,
okay.
I think for me, again, like having come from small company just launching into hopefully into the clinic next year, right? This we're in the meat of our CMC work for our lead asset, right? So master cell bank getting ready, the linker payload synthesis, everything sort of scaled up, and I think it's been relatively smooth for us, and I'll go back to because we start with really good linker chemistry, really good ADC design, like that. And Morris can weigh in, more specifically than I can on this point. But I think all of this, all of that, like what you start with, the foundation has been really critical to like the path of scale up and CMC. And to that point, right? Like I think there are things that obviously like my R&D guys didn't appreciate that, say, these larger scale CDMOs or more equipped CDMOs, when they scale up, like some of the optimizations that they think about, that they came up with to in our case, maybe like significantly reduce the amount of enzyme needed for specific reaction, or a certain synthesis step that we either got rid of, or purification that we got rid of, that we didn't appreciate because we just aren't at that scale, and that's not where our expertise lies. So I'd say that again, like starting with good chemistry is important. Getting to those sort of very technical but important aspects of scale up as quickly as you can will really help illuminate the what you have in the technology and the platform that you're working on, so that you can optimize, refine, switch a buffer, switch a column, switch a what have you, if you think that asset, your platform drug product is what you need at the end to unlock whatever biology or effect that you want to do. So I mean those are just some of the comments that I would echo in this regard.
Thank you, Michael.
So just to follow up on what Michael was saying is, I think in the recent experience we've had, one of the advantages we have is we're bringing forward a new platform, dual payload platform, and a lot of early chemistry and development was done in an academic setting, and it was developed a lot of different things were looked at. So our key scientists had a good idea of the development space. Then when we go to a contract manufacturer, they look at it from a different perspective, and it's good to bring in that different perspective because it addresses ability to scale up and focus on things that ultimately will improve the cost of goods. So it's good to synthesize those two approaches together.
Yeah. Thank you, Morris. I did want to get to some of these audience questions here. Before we do, Heather, would you mind posting the third and final poll question for our audience? Folks, we want to know how early you begin strategizing your CMC development and planning for a new ADC program. So while you respond to that, Morris, I had this question for you because it looks to be directed at CrossBridge Bio, but if you have dual payload technology, how do you design and control DAR and consistency and scale up? Is that something that you're currently in the throes of, or how would you respond to that?
Yeah. So one, there's different dual payload technologies. So there's going to be, different approaches, different chemistry to get there. But what they all have in common is when you what you end up with in the vial, you want to have a reproducible DAR, that's drug to antibody ratio for those that are new to this. And with dual payload or multi-payload, it will be defined as the DAR for payload 1 and the DAR for payload 2, and the same if there's a third and a fourth. So you have to develop analytics that allow you to characterize and quantify the drug to antibody ratio of each payload that you're putting on to the antibody. And ideally, what you want to be able to do is to have one assay platform to do that, because it simplifies things, rather than I'm going to use an ELISA to get the DAR off the first drug, and I'm going to use an HPLC to get the DAR off the second drug. You'd want to have one analytical technique that would deliver you in an easy and robust way all the DAR information, if you will.
Mm-hmm. That's great, Morris. Excuse me. I see another one here. If I have a marketing authorization approval for trastuzumab, what kind of manufacturing, analytical, and at a regulatory level should I take care to launch a biosimilar for Enhertu or Kadicla? Can you shed some light on that? Are you seeing the question that I'm reading in the chat?
I'm reading it now.
Yeah.
Sorry, I'm not fully understanding the
it doesn't
question.
sound like, I mean, it doesn't sound like it's an ADC, but maybe that's the point. I mean, maybe they're implying that trastuzumab-based ADC biosimilar, because that
That's right.
Yeah.
But however, one would say at the end, they're talking about biosimilar for Enhertu or Kadcyla. I do believe there will be biosimilars for ADCs, just as there have been biosimilars for non-conjugated antibodies or naked antibodies like Humira and so forth. There will be that. I think one could anticipate that creating a biosimilar to an ADC will be of an equal or greater challenge from an analytical or CMC perspective than what's transpired before with the naked antibodies, simply because the degree of heterogeneity is greater. You have the heterogeneity of the antibody, and then on top of that, you have some inherent heterogeneity associated with the DAR, the linker payload, and so forth.
Mm-hmm.
Yeah.
Agreed. And it would follow probably a similar regulatory path for other biosimilars in any case, right? So to bridge the data, biocomparability and the rest. So
yes,
it's going to be messy in terms of analytical exact specs, and data package could be messy. But
yeah.
But what I think I've seen, though, is some regulatory, and I'm not the expert on this, but some regulatory flexibility now on like what trials or not need to be done with a biosimilar. So I think the emphasis is going to be on the CMC aspects to make sure that tracks as best. Again, I don't know the specific package required, but I think recently I've seen that, right, on the clinical side, there's more flexibility there. So, but yeah, you'll need to make sure your data looks robust for that asset.
Yeah. And I did want to talk about, the regulatory aspect a little bit right towards the end, but one last audience question for now, from Michael, who asked, do you have any concerns or comments regarding molecular size, the large size of an ADC, a combination of proteins? Thinking about culturing, process intensification, harvesting, purification. Yeah, any thoughts there regarding like the actual physical size of these ADCs and how that impacts manufacturability? And if not, we could.
Well,
I just figured.
I have two comments about that. So first, in terms of manufacturability, the more complex the molecules get, one would anticipate there would be greater challenges with manufacturability. Now, that said, building off the antibody platform. Antibodies in general are very robust molecules. They can withstand a lot of different changes, if you will, and they still sort of work. Now, a completely different spin on this about the size of these molecules. There is a school of thought out there that antibodies, 150,000 Daltons, have difficulty penetrating large solid tumors. And there has been various technological innovations, nanobodies, smaller targeted delivery formats that have been tested preclinically. Some of this has come into the clinic. I personally think there's still a lot more potential innovation there to create targeting entities that are not the full-size IgG1 150,000 Dalton antibody, that you could make smaller ADCs, if you will.
Mm-hmm.
Just want to add to that. In terms of manufacturability for larger molecules, there are a set of analytical techniques and methods developed, but each year there are advancements, right, in mass spec technology, peptide mapping technology tools to quantify the conjugation sites, right, and there are, on the innovation part, there are also advances, AI-based characterization of protein sequences and antibody sequences to help with developability. So the field is learning, accumulating all this data. That being said, even regular antibody production could potentially fail, right? Anything could fail at any CDMO and CRO, and the success rate is pretty good right now. So if it approaches 90%, I would be very happy. But so far, it's been developed for large molecules pretty well.
And maybe one thing I'd add is again, and I'll keep pounding the table on this. It really starts with, the ADC is integrated, right? There's many parts and pieces here, so you have to think about it, collectively. And I can give some real world examples where we've gotten some questions from, potential partners thinking through our antibodies. We prefer them FC silent, and this removes the glycosylation site. And then there's been concerns about aggregation, related to that. So that's just even the antibody engineering. There's other mutations you can do, and that might impact, particular aspect. And then if you have a very hydrophobic payload, that could add an additional aspect. But you could potentially innovate on the linker to facilitate reduce the hydrophobicity of that payload, and then the overall hydrophobicity of the ADC. And then, on top of that, certain mutations, there's, I think, some recent data as well on the antibodies that can sort of help form a pocket that then might sort of reduce the exposure of some of the payloads that are hydrophobic, that then make it more hydrophilic. And then ultimately, that might make it easier to manufacture as you have, better physical chemical properties. So again, it's going to be very specific to that asset that you want to build, but you do need to think about all of those parts and pieces together when you're starting to ultimately say, this is the product I want to bring forward. So,
yeah. Thank you, Michael. I did see this interesting question coming through from Abigail, and it echoes some questions I saw in the registration from attendees. But Abigail says the lack of US-based CDMOs was mentioned earlier. For CDMOs or CMOs in the early stages of building out capacity, labs, manufacturing infrastructure in the United States, what competencies should be strengthened or focused on to ensure favorable service offering in regards to ADCs? If you're trying to transition from traditional parenterals to ADC manufacturing, what are the biggest and best ways a CDMO can meet those requirements?
One thing I'll mention, which we really haven't talked about, up until now, is if the question's getting at new CMOs setting up capacity in the United States, or actually wherever they are, new CMO, you want to make sure they develop really good quality systems, because ultimately you're making GMP product and it's for the clinic, and hopefully you'll do a commercial launch, you'll be a marketed drug, and you have to be able to do well in client-sponsored audits or regulatory affairs audits of the facilities, and that gets at the quality systems, quality assurance, and sometimes companies underinvest in that, and that can hurt them for a long period of time, if you will. So that's an important thing to add, in addition to, you know, being able to develop and scale up processes. You got to run them in the right quality environment.
In my view, I see a couple of major points come. I would, you know, first of all, there is a way to start with their smaller scale manufacturing to get sort of your foot in the door, you know, and establish all the processes as Borys mentioned, right? So you don't need to produce 1,000-liter scale fermenters, right? You could start with 50, 100-liter, 200-liter scale, at least for antibody, right? And basically this is one. And two, another idea is the regulatory support, expertise in early clinical stage development and regulatory support, knowing all the, you know, gray areas and all the rules, you know, that would really, you know, help clients, right? So that expertise is actually essential, in addition, of course, to, you know, CMC itself. So it's a regulatory CMC, I would say, very critical.
Well, I wanted to touch on regulatory, but unfortunately, we're closing in on the end of the hour here. So I just wanted to give everybody one last chance to speak directly to the audience in regards to CMC strategy and preparation around ADCs. What is your parting piece of advice or wisdom for our audience?
Michael, you want to go first?
I mean, I'll just echo the same thing. Really think about all of the pieces in an integrated fashion, right? Because again, if you talk about changing DAR, there was a question about like high DAR and, you know, manufacturability, volumes, concentrations, like all of these things. You know, a good linker might help reduce the hydrophilicity of a high DAR, you know, molecule, right? So like again, you got to just really think about all of these things, and you know, work with your CDMOs or your CMC group. You know, you guys work hand in hand to solve the challenges.
I would just like to say that this is a great time to be in the ADC space. It's had its ups and downs across the decades, but we've hit the tipping point. There's going to be a lot of great drugs coming forward in the next decade, a lot more innovation. So it's a good time to be in the space.
Thank you. Michael, Morris, Borys, thank you all for joining us today and sharing your wisdom with our audience. To those of you out there, I'm sorry we couldn't get to all your questions, but thank you so much for tuning in to today's Bioprocess Online live event. And thank you to Cytiva for sponsoring today's event. We'll see you next time.